So Dr. Jon Kobashigawa, welcome.
Thanks.
You know that this is part of the ISHLT history project that we've been doing for several years now. And the purpose of it is to have interviews with people that were instrumental or very important in the development of cardiac transplantation, heart-lung transplantation, and so on. And you're absolutely one of these people. And so we wanted to talk to you a little bit about your personal experience in the field, really starting from the beginning of time.
That's a long time ago, Jim.
Yeah. And so what we wanted to do, there are several questions that one thinks of when one looks at your kind of professional history. As I recall, you did really all of your training in California. Is that right?
Right. I started my internship at UCLA back in 1980 and completed my residency, then moved on to my cardiology fellowship at that time. I do have an interesting story, though, about that, Jim. I don't think you know that or anybody else really knows about this. So when I was an intern, I did my residency, and then I married this woman who was a very good friend of mine. We were great friends at Stanford. And then we both went to medical school in New York. She was at Columbia, I was at Sinai. And we were good friends, and we just happened to get together. And eventually we did get married in 1983. Well, it just so happened that her father was a cardiologist, and he had a very large cardiology practice in Los Angeles. Now, many people might think that I married my wife because of him, but actually we were very good friends even before in undergrad. So there was some expectation that I would graduate the cardiology fellowship program eventually and then join his practice. So everyone started talking to me about that, and I decided, I didn't like that. I was going to be a renegade. I was going to be the maverick here. So I decided on my own that after I do my residency that I would apply to pulmonary critical care. I liked critical care patients. So I actually took out applications for pulmonary medicine. And, and I asked the chief of cardiology, who I had done a rotation in the CCU, to be one of my references. And when I told him about it, he looked at me and had a very odd look and then said, okay.
Sort of like are you insane?
Are you serious?
Yeah.
So, I left, and then a few hours later, I was walking in the halls. He's walking toward me, he pulls me aside and said, John, are you crazy? You need to be in cardiology. You need to apply to a cardiology fellowship. By golly, your father-in-law's tremendous practice. Are you crazy? And basically he said to me, I'm offering you a spot in cardiology, which probably wasn't kosher because there was a match that was being done. But obviously he wanted me to apply to the cardiology fellowship. I said to him, well, you know what, let me think about it. I go home and it's Friday, so we have a family get-together, and I see my father-in-law. And I said, Nobi, my father-in-law, I said, Nobi, the chief cardiologist stopped me and basically offered a spot in cardiology. And I said to him, what do you think? He looked at me, smiled broadly, and said, I think you should take it. And then I realized they were all in cahoots about this. So basically, I figured it out, and I thought about it, and I said, I was probably being a renegade here, being a maverick, and I really should go into cardiology because I really did like critical care patients. And so I did. I applied and went through the usual interview process and with all the other applicants, and was accepted to the cardiology program. But that's how close it was, Jim, for me to be a pulmonary specialist instead of a heart specialist.
You never did go into private practice, though. You've been in academic medicine the whole time,
well,
is that right?
That's something, though, too. So, I actually joined my father-in-law in private practice. So for my 30 years at UCLA, after I graduated the fellowship program, I was in private practice. And so In private practice, we were literally the clinical arm of the Division of Cardiology. So we had, you might call it the best job in the world, because I had my own private practice, and yet I had academic medicine. So the chief put me in charge of heart transplantation in 1986, and we had done only 13 transplants by that time.
Now, tell me a little bit more about that, how you sort of segued into transplantation, especially you said this was in '83?
Yes, well, we began our program at UCLA in 1984.
So this was after cyclosporin was
That's correct,
introduced.
after cyclosporin came into
Right.
vogue. And as a fellow, I was intrigued, because that's the years I was a fellow. I was intrigued by that. And Lynn Stevenson and Hillel Lax, our surgeon at that time, were starting the program. And as a fellow, I was working with Lynn, and we were doing some research looking at waveforms, right ventricular waveforms, to see if we could detect rejection based on the change in time over pressure. But nonetheless, I got involved. And at that point, I took a fancy to transplants. We didn't do very much, just 13 in the first 2 years. And when I graduated the fellowship program, Lynn was not truly interested in transplantation, but more in developing her heart failure career. And so the chief of cardiology asked me to take over the heart transplant program as a private doctor. But the reason why that happened is because my father-in-law was very much a part of the UCLA family. Although he too was private practice, he was extremely well known, great financial expert, helped out the division, the School of Medicine, the chancellor. Didn't help that he donated a lot to the School of Medicine. He was more or less embraced by the full-time faculty. We even had a division within the Department of Medicine called the Division of Clinical Faculty Medicine. And my father-in-law and I were co-chiefs of that division. So as a division chief, I actually shared, the division meetings, division chief meetings, and so I was looked at as part of the full-time faculty, and everyone thought I was. That had some advantages, though, not being part of the full-time faculty, because I could do my own research. I had my own foundation. I developed my own foundation eventually after patients had donated monies to support my research. And so my interest in transplant stemmed from my early exposure to the early transplant patients that we had, and by, I guess, a stroke of opportunity and luck per se, I ended up as the program director in 1986. So we had done 13 transplants at that time, and we skyrocketed from there to the point where we were doing a high of 102 transplants in 1 year.
That's a big volume.
That's a big volume, and so we've done over 2,000 transplants altogether that I've taken care of. So it really worked quite well.
So this was, I guess, through the late 80s, early 90s, when you were really building a very significant program at that time. And then, now, when did you get, during this period, when did you get involved into the ISHLT?
Well, I began with the ISHLT actually back in 1984 when we opened our program, and as a fellow. I remember Lynn Stevenson saying, "John, we've got to go to the ISHLT," and I said, "ISHL what?" Anyway, it was actually the International Society for Heart Transplantation at that time. Not the L wasn't there yet. So I followed Lynn and up there, and she introduced me to this wonderful person named Sharon Hunt. And little did I know who Sharon Hunt was, but she was the maven of heart transplantation at Stanford. And so I remember meeting with Sharon, and just wonderful. She was absolutely wonderful. And I remember asking her a simple question, which I look back and say, God, she must have thought I was an idiot. I asked her, does cyclosporin really cause high blood pressure? And, Sharon was very nice. She said, John, actually it does. And, it's somewhat refractory at times. But anyway, it's just very gentle and kind. And I just remember that. And ever since that, we've been extremely good friends. And she certainly has been one of my mentors as I have grown in the transplant community.
Now, just to digress for a second there, since you were interacting with Sharon Hunt, around that time you must have known a little bit about Margaret Billingham's work then, because you mentioned earlier that you were looking at waveforms.
Right.
So then you must have found out a little bit later about some of Margaret's work.
Actually, Margaret's work in terms of diagnosing rejection and having that early 1990 biopsy grading scale was truly instrumental in one of my first projects that I had done, and that was with biopsy-proven rejection. This is back in 1986 now, and at that time it was very much understood that you had to give 1,000 milligrams of solumedrol for rejection, and you had to bring the patient to the hospital, admit them, and give them 1,000 milligrams daily for 3 days,
Right.
then they could be discharged home. So I was thrilled from the fact that we had a biopsy that Margaret Billingham had actually made a grading scale that we could say, yes, this is rejection. But I thought that we could do things better. Why? Because there were some reports that you didn't have to give 1,000 milligrams of solumedrol. You could just give prednisone pills and treat them with 100 milligrams of prednisone daily for 3 days, and that would take care of the rejection. So at that time I did a nice clinical trial based on Margaret's work and randomized 40 transplant patients who had rejection by her grading scale. And of course I could not have done this if Margaret had not done her grading scale and put that forth. And so I randomized these patients who had rejection on biopsy but were without obvious heart failure symptoms, and randomized them to prednisone, oral prednisone, or to the intravenous solumedrol, bringing them into the hospital. Yeah. Well, as it turns out, they were the same. It was the same. 92% of both sides had reversal of rejection, but there's quite a bit of cost savings. It took about $3,000 to $4,000 for the intravenous solumedrol, which was very cheap in today's dollars, but back in the 1986, that was a lot of money at that time. But the cost of an oral prednisone and bolus was $5.25. So, but that was my first introduction into clinical trials. Again, a lot due to Margaret Billingham's work in making the biopsy grading scale possible.
Well, you more or less ran with that kind of study, didn't you?
It was exciting. Yes, I kind of ran with that type of study, but from there I had realized that there wasn't a lot of clinical trials, meaning randomized clinical trials that were being done. And that was, and literally just about everything we were doing at that time was based on expert opinion and not due to real science. And that's what intrigued me and started me on the clinical trials pathways.
Well, you've been involved in a number of very important clinical trials. There's and some of them quite interesting. Of course, there's the one on statins, which is very interesting, and then the ones that you've done on tacrolimus and mycophenolate mofetil too. The statin story is kind of interesting. How did that come about? Well,
in the late 1980s, we had seen high cholesterol in most of our patients, and we were truly wanting to give the best care possible for our patients. And seeing this high cholesterol, we were under the impression at that time that lowering cholesterol was good. Now, this was before the big, randomized trials of cholesterol lowering with statins. The 4S trial wasn't published until 1995. And so we really didn't know whether or not statins were truly beneficial, but what we did know just from the other lipid trials that were there before us, the coronary drug project with niacin, for example, and the resin binder studies, were suggestive that lowering cholesterol was good to reduce coronary artery disease. And so when we saw the high cholesterol levels, we decided that we really wanted to do something about that. Now, I had known about the statins and how good they were, so we used Mevacor, which was lovastatin, one of the first statins. And once we started to use that in our patients, we started to notice that patients were getting muscle aches. And in 2 occasions, we had rhabdomyolysis, where there was just so much muscle breakdown, it caused kidney failure.
So you were using fairly high doses?
Yes, we were using,
Yeah.
20, 40 milligrams of Mevacor at that time.
Right.
And we did not know about the interactions. And once we started to see this, we wrote letters to the editor in the New England Journal and the other journals that something's going on with these statins, so beware. And then we started to look at it from a mechanistic standpoint. I started to look at other things, look at lovastatin blood levels, and in fact, they were elevated. There was an interaction between cyclosporin and statins. Well, make a long story short, I decided that I would do something else to lower cholesterol. And I'd learned that pravastatin was less likely to cause muscle aching because it was hydrophilic instead of lipophilic. So basically, I did a pilot study using pravastatin. It worked. There was basically virtually no myositis, no rhabdomyolysis. So at that point in time, I decided we need to do a randomized trial. Now, honestly, I wasn't smart enough to really know in advance that statins had any anti-inflammatory or what we might now call immunomodulatory effect. I just wanted to lower cholesterol, Jim. I didn't really, think about any other benefit. So I randomized 197 patients to either pravastatin or pravastatin. And at the end of 1 year, we had set up the endpoints, and I said to my colleagues, I said, it's time we took a look at the results here. And lo and behold, we had, yes, lowered cholesterol, but we had knocked down rejection. And I thought to myself, what is going on here?
That must have been shocking.
It was shocking to me. And I said, so I had my cardiology fellow at that time, and I said, Steve, do me a favor, do a literature search on statins and immunologic effects. So a couple hours later, he comes running back to me. He shows me an article that statins can knock out natural killer cell activity by 98%. And I said, wow, it's kind of interesting. We should really look at natural killer cell activity. So I went to Paul Terasaki, who had a great tissue typing laboratory. I asked Paul, could he work out an assay, look at natural killer cell activity? He said, sure. He had just developed his microwell technique that he could do this. So we did that, and long story short, looking at samples from the pravastatin-treated patients and the control patients, we indeed had knocked out natural killer cell activity. So, we published our work in the New England Journal, and so we were postulating a pleiotropic effect of statins, a cholesterol-independent effect, because when we looked at our cholesterol levels and the effects of the statin, it had no correlation to how low cholesterol was. In fact, it even worked in high cholesterol to knock out lower rejection incidence. And so that's when we realized we were really looking at some pleiotropic or cholesterol-independent effects. And, my colleagues took it to the basic science laboratory, and we took it further. We actually found that it worked because we were blocking HMG-CoA reductase enzyme up here, and you have all the pathways underneath, the geranyl farnesyl pathway, ubiquinone, cholesterol. And we actually followed the farnesyl pathway. And, we actually found some drugs that could block farnesyl pyrophosphate dehydrogenase, the rate-limiting enzyme. And we found a drug called parallel alcohol. It's from the orange peel. By golly, an orange peel. And we did some studies with rats and transplant rats, heart transplants in the rats, and we actually found that we knocked out rejection in those rats with parallel alcohol. It's not cyclosporin, not corticosteroids, it's just parallel alcohol, and we actually knocked out rejection in mice. Well, a lot of things work in mice, as we know, but now my colleagues have taken it to the pharmaceutical industry, and they're working on it right now. But we came full circle from doing a clinical trial, noticing and recognizing a clinical event, taking it backwards to the bench and looking at where we felt it might have benefit. Many other investigators have looked at it as well and have found similar results. Quite interestingly, 2 years later, the Germans presented their data with simvastatin. And as it turns out, their data was identical to ours. 2 programs completely unaware of what either one of us was doing. I'm so pleased I beat him to the punch.
That's perfect corroboration.
Yes. And so, we published our down in the New England Journal. They published theirs in Circulation 2 years later. But certainly it confirmed our findings that statins did have this immunomodulatory effect in the presence of cyclosporine.
So I guess we're up to about the 90s, and about that time then you must have been thinking about mycophenolate mofetil as well.
Yes. Mycophenolate mofetil was a new experimental drug at that time. We called it RS41663. I don't know if you remember those code words, but yeah, you can see it's indelibly etched in my brain here because those were the way we would say RS. We called it RS for short. And it was innovative, different mechanism, and truly represented a change in the paradigm of how we might treat our patients. And because it was more selective in terms of immunosuppression as opposed to azathioprine, which was like shotgunning and having problems with infection and bone marrow suppression. And so this was truly of great interest. So the company had a lot to do with it because they wanted to, Sandoz at that time, they wanted to start this clinical trial. So they brought many of us together. Now this was very important. Why? Because this was the first multi-center randomized trial in the field of heart transplantation. And this was in the early 90s that we were actually able to come together. And so this was exciting for me because, I was with the giants of that time. There was Dale Redland, John O'Connell, Les Miller. I mean, just great names in transplantation at that time. So we all came together and we hammered out a standardized protocol by which we would treat and randomize these patients. Then it came down to designating a steering committee and then a chair of the steering committee. Now, I don't know what I did wrong or right, but somehow I was voted to be the steering committee chair, basically the chair of this first randomized multi-center study, which I was actually, to tell you the truth, shocked about. But obviously I was very proud to accept. And so I was basically brought into the fold in terms of the network, you might say, in transplantation at that time. But it was really a great experience, and my colleagues there were just tremendous. And we were able to hammer out this protocol and to run with it. So this is over 500 patients that we randomized to mycophenolate or to azathioprine, both in combination with cyclosporine and corticosteroids. So we came out with a great finding that mycophenolate was better than azathioprine. And this is truly where we began in the field in terms of multi-center randomized trials.
Yeah, it's very interesting seeing that drug come into market and hearing the patient's perspective on that drug as well. Having spoken to one patient who works in our unit, and I said, well, do you like MMF more than azathioprine? She said, you'll have to pry it out of my dead cold hand.
Yeah, when you look at the clinical benefits of mycophenolate over aza, there's really truly no doubt. And what we all strive for is better efficacy and less side effects, and that drug truly fit that build.
And then you went on and did something similar with tacrolimus, is that right?
Yes, we ran through several multi-center trials thereafter, and tacrolimus was another one, another up-and-coming drug in the 1990s. Everyone was using tacrolimus for patients who had refractory rejection.
And at that time, it was called FK506.
Yes, FK506, another code word. You know, some people still call it FK, by the way.
Yes.
And so at that time, everyone was using FK by itself, and for refractory rejection. And you know what? It was working. It did well. And so in the 1990s, I thought, wow, we should probably use this drug, on our own. And at that time, I had approached the companies about that, Fujisawa, and they were they were sponsoring some multinational trials, first the U.S. trial and then the European trial. Dave Taylor did the U.S. trial, and Bruno Reichert did the European trial. But it was not with a standardized post-transplant protocol. They basically allowed everyone to do what they did. And the reason why is because it wasn't fully supported, and they could not support a standardized post-transplant protocol. So there are some questions as to whether or not it was truly a valid type study. So initially what I had done, I did a large trial in our own program of FK versus cyclosporin and showed benefit in a single standard protocol. And so I had mentioned that to the company, and that we really should do a good randomized trial with, again, cyclosporin and using mycophenolate, at that time. So this is now, it takes us into the 2000s, and by the mid-2000s we had actually put down a protocol. But with this protocol, we decided that we would add yet a third arm. So we had three arms. One was tacrolimus mycophenolate. The second arm was cyclosporin mycophenolate. But the third arm was, interestingly, tacrolimus and sirolimus, the proliferation signal inhibitor.
Yes.
Again, a different anti-proliferative, if you will. But we thought it was important to introduce that drug into the field because there was some
mechanism.
Right, unique
Yes.
mechanism, the fact that it might even decrease transplant corner disease. So we were able to do that in that three-arm trial, and we demonstrated that tacrolimus mycophenolate had significant benefits over the other two arms, especially cyclosporin and mycophenolate. And I think most of the field jumped onto that bandwagon, and now more programs are using tacrolimus and mycophenolate. And I truly believe it's that combination that really did work, and that really prompted outcomes benefit. Because when you look at the field in terms of rejection incidence, that has gone down, and that was demonstrated with the use of tacrolimus and mycophenolate as well. So there truly has been evidence-based medicine that has at least directed the field in terms of better outcomes.
Yeah, I could show you some very interesting time-related data related to immunosuppression use and infection and rejection. Well, I'll have to show you that sometime. Let's change gears a little bit. And what I wanted to do was to address your role in the ISHLT, because particularly in the late 90s, early 2000s is when you really assumed a senior role, a leadership role in the ISHLT. I recall that in 2000 you were the program chair, and in 2003, 2004 you were president. Now, when you were program chair in 2000, that was an interesting meeting. That one was in Osaka.
Yes.
It's the one and only time we've had a meeting in Asia, and in my memory, it was one of the most interesting meetings we've had. Can you tell us a little bit about how that came about and how the meeting came to be in Japan at that time, and what led up to that decision?
Well, I had been very interested with the ISHLT, working on various committees and the like. So I was getting to be, I suppose, more well known in that light. So Bob Cormos, president-elect, the year before, gives me a call, out of the blue, and Bob says, "John, how you doing?" And I go, "I'm fine, Bob." I'm wondering, why is Bob Cormos calling me here? And he says, "John, I want you to be my program chair." And I said, I was in shock. I said, "Bob, let me think about it." So Bob says, "John, are you crazy? I'm asking you to be program chair. This is an honor." So Bob was just amazed that I would even say, "Let me think about it." And Bob just said to me, "John, you would be perfect. We know it was already determined at that point that the ISHLT would be in Japan." Now, I had been on the board at that time, and I'd been through the discussions of Japan, so we knew that there was great interest in going to Japan because Japan had just passed brain death laws, and so they were truly interested in having the ISHLT come so that they can expand donor awareness. So it was truly the right time at that point, that period, to go to Japan. And there was, so I said finally yes to Bob, and so we started to get excited. And I think Bob thought I spoke Japanese fluently, but I actually don't. I'm actually third generation Japanese, and so my Japanese is very limited. But I think Bob was okay with that at the end of the day. In formulating the ISHLT in Osaka, they had just built a big, very large convention center in Osaka. So the timing was going to be crucial as to whether or not they would finish it in order for us to be there. And in fact, we were the very first group to use that new convention center in Osaka, and everything actually worked well. There was an Osaka or Japan organizing committee led by Dr. Kawashima, who is one of the senior surgeons in Japan. They were wonderful to work with. And they put together, with us, a tremendous program. We put together the scientific content. They actually put together a welcoming ceremony and the gala, which was absolutely phenomenal.
The gala was
the most
interesting gala I've seen.
They had traditional kabuki dancing.
Yes.
I mean, the culture that was there was just, and geisha, women that came around. It was absolutely phenomenal.
That one remains my favorite.
Absolutely, and then breaking the sake barrels. I remember myself and Bob and Ann Keogh, the previous president, we were all breaking these sake barrels to open them. It was quite a tradition. We learned a lot about Japanese culture and history, and actually it was good for me too. I learned a lot as well. But it was a very memorable experience. The science, as always, was outstanding. And many of my colleagues truly remember that as well.
So after that unique meeting then, a couple of years later, you became president of the ISHLT, and this was in 2003, 2004.
Yes, Les Miller called me, and Les had been one of my longtime friends, and he said, John, the board met, and we'd like you to be president. And I was, again, shocked. I had no idea this was coming my way, my direction, and I'm sure my mentor too had something to do with it, Sharon Hunt as well. She always smiled at me, before, and I was figuring this out that there was something going on. But nonetheless, I was very honored to take on the presidency at that time. It was tremendous. Being program chair, I had truly gotten into the mainstream and the nuts and bolts of the society, because as program chair, you actually work harder than the president,
yes,
trying to get things done together and maintain connections and networking, because that's what the program chair does, which was great for me because it was a great segue into the presidency, because I pretty much knew, how things were working, and just rely on Amanda Rowe, by the way, to help me through and navigate through all of the administrative duties of the president. So it was quite a challenge and enjoyable at that time. We were just getting into mechanical assist devices. And as president, we were formulating the registry and who would take it on at that time. So I was working with a lot of the companies, the device companies, and working with Jim Kirkland to formulate a strategic plan by which the ISHLT would be the one to be involved in mechanical assist devices. When you look back upon that time, that was a turning point for the society. And why? Because when you look around today, the ISHLT has taken a role in mechanical assist devices. I mean, how incredible that was when you think about it. Because devices, heart devices, have expanded tremendously. And in fact, it's exploded on the scene. And to have the ISHLT, the society by which everyone goes to in terms of registry, in terms of technology, it's second to none. And to have that foresight, to know that this area would truly be almost a society by itself in terms of its importance and its expansion, is truly incredible for the society. So we didn't know that at that time, Jim, but obviously, with the leadership afterwards, it's been a godsend for the society to expand to these areas of research and new device technology.
Would you consider that to be the most critical issue when you were president of ISHLT, perhaps in hindsight?
Well,
or
yes.
were there other issues that really concerned you at the time?
That was one of the issues. One of the other issues as president, I also was determined to redo the ISHLT grading scale, because at that time, antibody-mediated rejection was not even accepted. And so I organized the consensus conference on revising the biopsy grading scale.
And that was very important considering that the previous effort that we know of in the 90s did not succeed.
Did not recognize antibody-mediated rejection.
Yes.
And so when I put that together, I had I had placed Susan Stewart as chair of the pathology council to chair it. And so we organize the meeting and we get there, and I'm expecting Susan to run it. And Susan looks at me and says, "Jon?" And so I end up running the consensus conference meeting. And little did I know that I had stepped into a quagmire of conflict. There were people there that were adamant that antibody-mediated rejection did not exist. And the tempers that were flaring, the arguments that arose, people threatening to leave the room if they were not heard or their way was not looked at. I was actually appalled by that. And I was saying, oh my God, what am I going to do here? And over time, we were able to hammer this out. Basically, the mantra was, can you live with that? And so, as I was sharing this and saying to everyone else, can you live with this? And the bottom line was, yes, we can live with this. But also I said to them too, this is not the end all. We'll be reviewing this and revising this biopsy schedule again and again, and we have just a few years ago. But and after people realized that this was not going to be the final grading scale, that it would be revised again and again and again, that people realized that we needed to have some platform to view antibodies. And so it was finally passed, but not without a struggle.
And you've had some subsequent consensus conferences after that.
Yes, antibodies have always been an area of interest to me. So I had organized two other consensus conferences. The second one that I did after the revising the biopsy grading scale was on sensitization, antibodies in patients awaiting heart transplant. And that worked out quite well. We came up with a great white paper, consensus conference paper, to approach that problem. Then I did a second one with antibody-mediated rejection, and that worked out quite well. That was 2 years after the sensitization conference. I actually did another one after that with nursing, and that was wonderful to actually pay tribute to our nursing colleagues. That was crucial conversations in transplant nursing, which was truly fabulous. And then now the one we had just yesterday, our consensus conference on primary graft dysfunction. So we were able to hammer out a definition and grading scale, which will be the ISHLT grading scale of primary graft dysfunction. So quite satisfying, to say the least, in terms of moving the field forward. But it's not only me. I work with great people at the ISHLT, and one cannot do things alone. You have to have the camaraderie, support, and vision of your colleagues in order to do this. And so certainly I pay tribute to many of my colleagues here, and many of the young colleagues that are the future, and we've included them as well.
Well, why don't we wrap up with one question I had, and you've actually recently published a paper on this, on the future of transplantation as of the year 2013. I'm curious what your opinions are on that.
Well, initially when Alan Kirk asked me to write that for the AJT, it was to see where we are today in transplantation and whether or not there was doom and gloom for heart transplantation in the future. When in actuality, in reality, I think there is a great future for heart transplantation, especially with new technologies, better understanding of antibodies, and genomics. There'll be customized, personalized medicine, as we now call it, where we can customize immunosuppression for our patients. And again, a great deal of exciting work having to do with tolerance. Here I am talking to one of the world's experts in immunology right in front of me, Jim, and as you know, there are many exciting areas to look at, whether it's bone marrow transplant along with a heart, maybe some facilitator cells that are being used to encourage acquired tolerance. All these things may occur so that one day we will not have to take immunosuppressive agents. The body will accept the donor heart as it is. In that situation, truly, everything else will be secondary to this new form or new wave of heart transplantation. Total artificial hearts may be there in the future, but that's way down the future in terms of taking heart off the shelf and putting it in and not worrying about the battery life, et cetera, and anticoagulation. So I think it was a challenge, but very exciting for me to write that article and to see where we are and to sit back and say, wow, a lot of things have happened in the past, but yet the future is even brighter.
Well, Dr. Kobashigawa, thank you very much for sharing your history and your opinions with us.
Thank you so much, Jim. I enjoyed it.